And I will now hand over to Bevan. And I just realised I don't know if we're going to pronounce it Biotins or Biota NZ. So I guess you can tell us about how we're going to be able to access name and literature data on New Zealand fungi. So thanks very much, Bevan.
Helps if I unmute.
Yep.
Yeah, that was one of our kind of questions. I'm going for Biota NZ with a nice pause there. Yeah, so a bit of a different talk now. So this is not a talk about science research, but rather about our databases and by extension collections at Manaki Whenua that enable some research to happen. So we've got this new website, Biota NZ, and although this is a new website, the underlying data is the same. So the data that feeds in is the NZ fungi database. But before we get there, it's kind of worth thinking about different kinds of databases. And I know I haven't fully shared my maximize screen. That's for a reason, because I'll be flipping back and forth between a web browser. So before we get into Biota NZ or NZ Fungi, let's think about what is a collection database. So, well, I was last year we were on Stewart Island, and this is one of the fungi that I collected at the time. So what I called Coprinopsis lagopus, although I hear from Jerry, it's actually 3 different species now, typical. Just one, that I could easily identify. Now I can't. Anyway, so here I have collected this little ink cap mushroom. And in the background, we see my piece of paper. We've scrolled down the name that I think it is, the date, where it's from, who collected it, who determined it, the locality. So basic sort of data that we need around a specimen. Now, I probably should have put a lat long in there, but I use a sort of a GPS to get that and add it in later. And this is really sort of the basic data that you have when you're collecting a specimen. So if we think about the workflow from here, so we're out in the field, hopefully like we were doing this week, we collect a specimen, and then we have to make some kind of notes about it. For the foray, we have these really nice pieces of paper labels that we can write things on, or that might go into a notebook or a physical or electronic notebook. And then there's the process, if it's going to one of the collections, say PDD, for example, it needs to be physically accessioned. So that means that mushroom needs to go physically into the collection. And has quite a few steps to it. And particularly in the case of this ink cap mushroom, it needs to be dried pretty quickly before it all melts away into the ink. So you could dry it, and certainly in PDD, then there has to go through several steps, get frozen for a couple of weeks to make sure that there's no insects on it. It needs to be carefully packaged in acid-free paper inside another package. If it's a large one, it might go inside a box. So it's kind of like the physical curation and the physical accessioning of the specimen. But then you've got the same thing to do with the data as well. So we've got to get this data off your piece of paper or your notebook into an electronic form. And so that would be data accessioning. So putting the data into some kind of database. And that data could be different forms. So we could be transcribing the label data exactly, like here, or could be different things as well. There could be a photo or DNA sequences. So these would then go into a kind of a local database, and this might be in different forms. It could even be an Excel sheet at some very basic level. At Manaki Whenua, we have relatively sophisticated databases that are sort of custom made, but there's other ones around the world as well that are used, to great effect. And this would be typically locally accessible, so on your PC or within your institutional network. A next step further is to put it into a web database. And so this might be available on the internet, that people can look at that data and see. And then there's yet another step again, which is making that data into a federated database. So that might be going externally out to MyCoPortal or to GBIF, which will be available by others. So if you can note that each of these steps, there's sort of more data accrued, and the ability to use this data also increases as you get increased levels of digitization. So if we just think about what actually goes in there, so we've got this the taxon name, so the name of the fungus, the location, date, who collected it, who identified it, hopefully photos, really important, fresh ones, or maybe back in the lab, ones who've dried with a scale, maybe for photos of microscopy or spores. Maybe ideally, in many cases, we might have a DNA sequence, particularly for the newer collections. And perhaps if it's part of another study, we might have some morphometric data and maybe even chemical data. For example, some of the xylariaceous fungi, you can use chemical extraction, look at the extracts of those. And potentially also down the stream once it's been used in literature. So that kind of all makes sense, right? So specimen database is kind of like that label, that piece of paper, but made digital. And because it's digital, we can add in a lot more. And the idea is this thing called the extended specimen, which is the extra stuff that's not just on the labels. That's photos and DNA sequences and morphometric and that sort of thing. So before I get into BioDNZ, I'm just going to go through our specimen database, which is the SCD. And I'm doing this because this still exists. This will not be replaced at all. So here's the SCD page for a culture. And this happens to be the culture of Phytophthora acidicida, the disease that causes cowled dieback. And it's the holotype, or the X holotype culture. So you might have some photos, and these photos are specifically only of this culture. And then you've got a bunch of data that says, well, this is its barcode or its session number, it's a holotype. Usefully, you know, when was the database last updated and when it was added. We have this concept of components, and this is maybe not too intuitive, but if you think of it like it sort of came from the herbaria. So if you think of it like physical components, maybe in a specimen packet or on a sheet, these would be the different parts of it. Now, for us in the fungi world, some of those components are not present. This is kind of a confusing thing, and which would you call the host. So once you kind of think about it as components of a specimen sheet, it sort of somewhat makes sense. And this is what we do, because at Manaka FUN we've got the insect, plant, and fungal and culture collection. So we kind of have to compromise a bit on each other. And this is what we've got. So components. So we've got the active component, which is the fungus itself. And these are different identifications throughout time. So here's something before I wrote the paper, which I just called it Phytophthora species taxon agathis. And here we have the other component. Confusing, but this is really just the host that it was found on. Now, this does kind of make sense in some scenarios. For example, you might have a piece of wood that has a discomycete on it, a little pleurotus, and maybe a lichen on it as well. And so the host wood, maybe that was agathis, let's say, and those three species, they'd all be different components. It's just that often our host component isn't actually physically there. And in the case of the culture collection, really shouldn't be, because it should be a pure culture. And then we go back to this primary stuff. So this is the collection event, where and when it was collected, all that sort of thing. We've also got these extra things, the culture flags. So this is happens to be an unwanted organism, so it's got various restrictions put on it, which means we can't just send it out to everyone. They'll need a specific permit to have this, and various other custom notes. In this case, for the culture, and it could happen with the PDD specimens as well, we've got a full genome sequence. If you click this will link out to GenBank. Here's a bunch of gene sequences that I did, and this is a mitochondrial genome as well. And we've also got information on literature. So where this exact culture has been used in literature. So there's obviously the one where I described it in the genome paper and some others. And then because it's a culture, we've got a little bit of extra information about where it's isolated from. So all up, this is our specimen database. It's everything about an individual specimen or a culture. Okay, so we've got a specimen database. What about names databases? Now there's some out there, and some pretty major ones. So you might have used Mycobank or Index Fungorum. And these are websites that some of them have sort of expanded in their scope, but they started as nomenclators. So really just a list of every possible fungus name out there. A similar one in the insect world or for animals is Zoobank. And both Mycobank and Index Fungorum and Zoobank, these acquire all new names because somewhat recently all registered fungus and animal names require registration. So formal process of going onto the website and registering a unique series of digits for that name. For plants, they're perhaps a little bit behind, just clicking around the place. And so there's no official list of, well, there's no registration for plant names. If you're really into it, I find Tropicos pretty good for global plants, and of course, Biota NZ for the New Zealand plants. For bacteria, again, there's nothing official, but the LPSN site is sort of almost a de facto official site. And for all of these, you can plug in a name of whatever organism you're interested, get the correct spelling back, and some detail on it. But these don't necessarily, in fact, as a nomenclator, they would not necessarily do synonymy. So they might not tell you what the latest name is. They're really just a fancy list of names. Okay, so let's go to FUNNZ Fungi database. And so we'll just power through. So this is the first one, and this was developed by Jerry Cooper more than 20 years ago now. And it does all of these things. So what it does is it takes a specimen database and a names database and also a database of literature, so all sorts of publications, and puts it together and derives something that's more than sort of the sum of its parts. So you can do things by mixing together names, specimens, and literature. This was then replaced by this, which you'd probably be quite familiar with, which is the FUNNZ Fungi 2 website. So what's wrong with these? Well, actually, nothing. Nothing's really wrong with them. The data underlying is still good. It's just that there are issues with old websites and old computer code, and particularly around security. And so these websites and the underlying servers are just unable to be maintained anymore. So we needed to build a new website. And in doing that, we're able to improve on some things, and maybe not all things are improvement. So this is the new website. It's called Biota NZ. Do not call it Biotans, as I was tempted to when I first saw that URL. So let's have a look at a page. So here we are looking at the page for the taxon, so the name, Phytophthora agadicida, the disease of coldy trees. And we're just going to go kind of somewhat slowly through this first one, and I'll show you what all these bits mean. So here, and you can hover over these little icons and they'll tell you what you mean. So this is the a name page as opposed to a literature page. So it's a little alpha Linnaeus, little name page. This is a fungal name, and this is a really important point because Biota NZ merges together the fungi, bacteria, plant, and invertebrate collection databases. So this is very important to know that you're on the fungus or the NZ fungi page. This is New Zealand relevant, and that just means we've ticked a box somewhere in there that's relevant to New Zealand, and as the thing killing kauri trees, you could certainly argue that it is. It's a current name, so that's quite important, that it's not a synonym, and also that this page, you'll see, has information on collections and also descriptions. So up top is our image gallery, and so you can kind of click your way through here, or you can just hit this button. And what this image gallery doing is pulling together every image from every specimen. So it's collating all of those specimen databases for each of these different cultures and pulling it all together. And as you can see, we've got some spore pictures and some sporangia and a whole bunch of culture photos. Now, each of these will have a particular different description. What we have not got incorporated yet is the license information. So although the copyright linker research, many of them will be CC by 4, which is a free license that you can use, but that's not explicitly stated yet, unfortunately. So if we keep going down, we've got details on the scientific name, the reference to it, so you can click out to that. This biostatus is quite important, and I'll go a little bit later through how this is derived. But really, this is saying, is this thing present in New Zealand or not? And what is its origin? So is it endemic, exotic, etc.? Often there'll be some kind of comment, because this is a hypothesis, essentially. And so here's me, and so we'll often have one of us, PRJ for Peter, GAC for Jerry, and a date saying something about how we derive that conclusion. And for me, I'm kind of equivocating here because I say it's kind of uncertain whether or not this is an endemic species or not, but I am kind of leaning on the side that it's exotic. Time is progressing, so kind of powered through. So we've got a whole lot of stuff about the nomenclature, which will be interesting to some people. For example, the etymology, how we derived this name, the classification, vernacular name. So it was called PTA at one point. And this is really important to the associations data. So we've got this pool, so this database pulls together everything. So from my paper, I've mentioned that it was a pathogen of Caldi. And it's also noted that there are 72 isolates in ICMP, and every single one of them was from Acantharis australis, and also that one in PDD. This is a very, very simple scenario. They're all from a single host. And I'll just power through this. So this is just a description pulled from the paper, lots of detail there. Taxonomic concepts, we'll go through that, something in a different example. And this is something that's possibly a slight regression, but this is a list of all the collections. And under the old FUNNZ Fungi 2 website, every single one was listed in a massive table. In this one, we've collated them together because to kind of make it work as a single page and to fit on a cell phone screen and all that sort of thing. So for example, there's 37 ICMP cultures from Northland. And if you click on that link, it'll take you out to that SCD website we've just seen before, and it'll list all 37 of those. So let's look at a mushroom page. And so this is, I'm sure you want to be very familiar with this species. And again, we can look at the image gallery and have a look at all the different photos that have been taken over time, including some microscopy ones, field photos, some nice lab photos, various other notes that are pulled from the specimen packet, drawings of things. So it's really just lumping everything together. Same kind of thing, but what I want to go down here for is the host. So now you can see that there is a lot of different hosts listed and where they derive from. So these top two derive from this Gadgil book, and this publication here from Eric and Peter, and also from all of these PDD records as well. I'll go through that in more detail in a minute, but what I wanted to show you here is that there's different descriptions. And so if we go back in time, down the bottom, this is Greta Stevenson's original description of this mushroom, and you can read it all there. And then a later description by McNabb in his Bolotacea publication. And then another kind of description by Peter Gadgil. And then some notes in an unpublished manuscript by Jerry when he went to Kew and saw the actual type specimen. So there could well be, in cases many are, different descriptions in here. And for some of them, they might have a short description that was derived from, say, our NZ fungi book. I'm going to power through because I'm running out of time, but as well as pages on individual species, there's a page for publication. So this is a publication that I have, and it's listing everything in there, including descriptions that are derived from and all the scientific names that were mentioned in that publication. Okay. How do I use FUNNZ Fungi? Well, I use it to check names and biostatus, but one of the big things that I do is I use it to try to figure out what things are. So this is a specimen that I collected on Monday, and this is a bacterial leaf spot of Hoheria. And I know it's a bacterium because the leaf spots are quite distinctive. So they've got these sort of angular margins following the leaf veins and this kind of chlorotic yellow halo around it. So I know it's a bacteria, and I know it's from Hoheria. Now what? So I can type in Hoheria into here, and there's two options here. I can search all data, or I can search associations. So I'll search associations for a start, and we can restrict it to bacteria here. And so this is searching. So for each of these bacterial species, it has been isolated from Hoheria in the past. No time to go through all the details, but I kind of know that these ones were from me and Rose, I think, isolating from flowers, so from nectar. So they're not pathogens. So the very likely pathogen is the Pseudomonas syringae and this Xanthomonas campestris. So that's a pretty good start. The other way is to just search all of the data. So Hoheria, and this is kind of worth going through in some detail here as well. The FUNNZ Fungi database is 40,000 names, and this is also searching databases as well. So there's a lot that you need to kind of filter down. But my hint is to change this over here and go to relevance rather than title. And I've suggested to the database developers that this become the default, because look, right up the top is Hoheria. You can also use all of these things down here to filter in various ways. If you're just interested in fungi, then clicking on names fungi, which would probably be a good idea. Or if you want to look at the Hoheria and find out what it is in the plants name database, you could do it there. But anyway, we're going here, Hoheria, and notice that with the little fungus, so we're in the fungal record. Now, this is an improvement from FUNNZ Fungi 2, because in FUNNZ Fungi 1, shall we say, you were able to look at a plant genus and look at all the subordinate species, but you couldn't do that in FUNNZ Fungi 2, but you can in BioDNZ. So what we can do, this is Hoheria. So we're now looking at the page for a plant name. So if we go down to the associations, we can see here all the associations. So this is everything that was found on Hoheria. And this is a kind of a truncated part of it. So we can click on full associations. And this is it. And this is long. And I'll just skim down. And we can have a look. Oh, look, here's a leaf spot. Oh no, but that's a fungus. That can't be that thing. Okay, here we go. So we've got some blank sunken lesions with magenta halos. Well, they're magenta, but they're certainly angular leaf spots. So very likely my thing is Pseudomonas syringae on this leaf. So I very often use that kind of association data to at least get me started in terms of an identification if you know your host. And this will work for all of the mushrooms and plant diseases as well. I'll just skim through this really quickly. So you can also look at vernacular names. So I've just typed in a roundhead here, and we can look at blue roundhead, conifer roundhead, and if you click that, it'll take you out to what the species name is. Just another kind of hint in terms of searching. It's not like Google in that it will kind of guess if you spell something wrong. If you spell something wrong, it's not going to work. So you might have to try variants, or what you can do is just add this wildcard. So this, and particularly if in case I'm not sure if a mushroom is a Coprinus or a Coprinopsis, I can just put Coprin and a little asterisk, and that'll pull up all those different names. So that's a good hint if you're, because it doesn't automatically do substring searching. If you can also put the little asterisk in the middle of the word to search for any kind of internal variants as well. And if you want to know how to do it, yeah. So if you look at these about things down to search syntax, it'll tell you all in great detail about how you're able to search things. Right. Okay, we did that. I'm nearly done. I just want to talk about the biostatus. So this is a screenshot of internally what it looks like to us. So you can have absent and present, but we can also have various subcategories of these. So you could have absent, but absent because it was eradicated. So you'd click that little box there, that radio button. Present, maybe something's only present in captivity or culture, you know, some kind of cultivated mushroom, perhaps, could be that. So it might be present or absent, but there could be some more detailed version. Exotic is pretty obvious. Now, this is a probably important one. So endemic means found naturally only in New Zealand, and indigenous or non-endemic, essentially the same thing, would be, say, if something was in Australia and New Zealand, but that's indigenous or non-endemic to both countries. So that's just a way of interpreting the biostatus data. And yeah, so kind of a very, very quick overview, but try it out. Give it a go with your favorite fungus or bacteriochromis, just bung in the name and see what comes up. Does it make sense to you? Yeah, let us know of any errors or clarifications and suggest any improvements. And yeah, you can email me, or at the bottom of the page is the Biota NZ page. Very happy to take any questions now.
Right. Thank you. That's really good. I'll let you go on and on because we use it every day, really, your databases. It's so immensely useful. And some of the comments in the chat have been saying the same thing. Yep, Teresa says it's very cool. She uses it a lot. Nicola Day, hi Nicola. She said that the associations option is so helpful. David White, you can read these here yourself anyway, suggesting that we could use a conversion for languages. And Anna really wants to look at seasonality and is suggesting you could make your tables bigger and busier just so she can do that. So what I might let you do, if this is okay, just because it would be good to get done by 5, is let you deal with the comments there if you like in the chat.
Yep.
So thank you, really good. There's another one there too. Kieran, yep, look, I'll let you handle those.
Yeah,
so thanks very much,
Thanks.
Bevan. It was fantastic.